Thank you, Phyllis, so much. That was a very informative talk. I hope we all learned a little bit more about, vaccine development and production and things that we can do for ourselves. This talk, however, is going to be about sort of mRNA vaccine production and the science behind it. And it's going to be starring Dr. Weissman. So Dr. Weissman is a professor of medicine at the Perelman School of Medicine at the University of Pennsylvania. He received his undergraduate and master's degree in biochemistry from Brandeis and his MD PhD from Boston University. Dr. Weissman's laboratory focuses on the study of RNA and innate immune system biology and the application of these findings to vaccine research and gene therapy. Almost 23 years ago, Dr. Weissman began studying how messenger RNA, otherwise known as mRNA, could be used to create vaccines. And now his mRNA research underpins two of the leading coronavirus vaccines, which is the Pfizer and BioNTech vaccine and the Moderna vaccine. Dr. Weissman, if you're able to turn on your screen, thank you very much for being here. Would you mind also sharing your slides if you have them? I believe you have permission to do so.
All set?
Yep, we're good.
So what I wanted to talk to you about is really to give you a background on vaccines in general, and then finish up with mRNA vaccines. This is important because we're in a world with COVID-19 where vaccines have taken the forefront in the potential treatment, elimination of this pandemic. I have a couple of disclosures. We've got patents, et cetera. So let me just start with giving you an idea on involved in starting to make a vaccine. And the first thing you do is you concentrate on the target. In this case, it's the SARS-CoV-2. That's the virus that causes COVID-19. This virus has a number of different envelope proteins. So those are proteins that are on the surface of the virus. The virus uses these for a number of activities. In particular, the spike protein is the protein that binds to our lung cells, our respiratory cells, and mediates infection. The protein that does this spike is interesting because what it actually does is it exists in multiple conformations. In one case, the receptor binding domain, so the piece of the spike that binds to the cell and infects, can be hidden or it can be exposed. The virus likely does this to avoid immune recognition. So it only pops up its receptor binding domain when it wants to infect the cell. That gives you the basis for SARS-CoV-2 vaccines, that is the spike protein. There are many different platforms, approaches that have been used to develop vaccines against the spike protein. These include inactivated or live virus vectors, viral vectors, DNA, RNA, and protein subunits. As you can see, there are numerous clinical trials. There's about 60 ongoing clinical trials right now for COVID-19 vaccines, but there's numerous in each group. This gives you a sort of a broad overview of vaccines in general. The first vaccine that was ever made was against smallpox. And it's actually an interesting story. Edward Jenner was a physician in the UK. And during the smallpox pandemic, he noticed that milkmaids didn't develop smallpox. He also noticed that the cows that they worked on had similar looking pox lesions. So what he did is he broke open those lesions and injected the contents of those lesions into people as a vaccine. What he was actually doing is that cows have something called cowpox that's related but not infectious for humans. And that was a attenuated pathogen. So it shared the ability to make an immune response that could protect, but it didn't infect most people. Inactivated pathogens, including things like typhoid, cholera, and a bunch of others, were developed over 100 years ago. These involve typically the influenza vaccine that most of us receive, where influenza is grown in chicken eggs, it's inactivated and injected into people's arms. Protein-based vaccines, where they take a single protein from the virus or bacteria, produce it and inject it. The tetanus toxoid vaccine is the classic. Newer platforms include viral vector. Here, what researchers do is they take a common cold virus like adenovirus, and they insert into it the protein they want to make an immune response against, in this case, spike. DNA and RNA, other genetic vaccines, and we'll describe all of these in more detail. So when you start thinking about developing a vaccine, we turn to the WHO, and they elicit guidelines for how to make a good vaccine for COVID-19. And what they identified are 6 issues. So the first is safety. And what they're looking for is a vaccine that gives mild, transient adverse events, is well tolerated by all ages. This is everything from young infants through elderly adults, and has good potency. Efficacy is important, and it's important for 2 reasons. One is it has to be efficacious in general, and they say at least 70%. But importantly, it has to be efficacious in elderly populations. Many vaccines suffer efficacy in the elderly. They want something that's stable, that can be stored at higher temperatures, preferably in the fridge or at room temperature. They don't care about the route administration. They would prefer non-parenteral. What that means is not through an injection, such as oral or nasal. For doses, they prefer a single dose, but would accept 2. And the vaccine also needs to be able to produce on a large scale and be affordable for low and middle income countries. So this gives a general overview of what we look for in a vaccine. And what that involves is we want something, so the way a vaccine works is that you deliver an inactivated virus, a genetic code that codes for the antigen of interest, in this case, spike. That vaccine is presented to the immune system. It elicits both antiviral cytokines. These are proteins produced by a cell that help to kill or neutralize. It produces antibodies that can directly bind to the virus and inactivate it, and they induce cell-mediated responses, in this case, T cells, that recognize an infected cell and kill it. So let's go through the different SARS vaccines that are currently in development and discuss them. So we're going to start with an activated or live virus vaccine. The heritage of these vaccines is that they date back over 200 years. The smallpox was the first attenuated vaccine ever used. Many viruses currently in use fall into this category: measles, mumps, rubella, rotavirus, and others. The immunogenicity, so their ability to induce long-lasting humoral and cell-mediated immunity, is very good. The safety profile is good. The main risk is that sometimes, and this occurs incredibly rarely, the attenuated virus can become more virulent. Uh, One of the drawbacks is that since you're growing a live virus, you need dedicated biosafety facilities. It's often given by oral, nasal, or parenteral IM injections. These vaccines are slow to develop. So in order to attenuate a virus, it often has to be grown for long periods of time in culture. It's also difficult to scale up. So to make many doses of the vaccine requires very large tanks of cells that produce the virus. It also can be fragile and impaired by heat and light, typically needs to be frozen or refrigerated. The mechanism, this would be a SARS-CoV-2. What you do is you grow SARS-CoV-2, the virus of COVID-19, in cells, um, collect large amounts of the virus and either attenuate it or inactivate it. There's many ways of inactivating the virus. When that virus sees a human cell, it enters, its proteins are then processed and presented to the immune system. If the virus is alive, like smallpox or MMR, the virus begins to replicate but can't complete infection. So it doesn't spread to other cells. But as part of that growth, it produces more protein that gives long-lasting immunity. The next are inactivated pathogens. So this dates back over 120 years with things like typhoid, polio, rabies, and others. The immunogenicity of these vaccines is often somewhat limited. In other words, a live virus gives very good immune responses. Killed viruses, pathogens, are less able to produce strong immune responses. They're typically very safe, but again, they require the handling of an infectious virus. They're typically given by the intramuscular route with usually at least 2 doses. They again are slow to develop. It takes a long time to grow the virus up, to figure out how to inactivate it. But they tend to have good stability, so they can be transported at regular fridge temperatures. Their mechanism of action is they essentially enter a cell, the cell processes them and presents them to the immune system. Protein subunit vaccines are again very common. They date back over 100 years. The tetanus toxoid is the classic protein subunit vaccine. There's many approved examples that are commonly used for hepatitis B, HPV, and others. Their immunogenicity is often limited, and that's why researchers and pharmaceutical companies will use adjuvant. An adjuvant stimulates the immune system to make a protein vaccine more potent. Its preparation is not difficult, but again, it involves cells producing the protein, which requires extensive facilities, and it means having to add an adjuvant. The adjuvants are known to cause inflammation and in some people allergic reactions. They're typically given by the intramuscular route and often 2, 3, or more doses are required. Their development is moderate. It's shorter than live virus attenuated vaccines. You still have to have cells that produce the protein. You have to collect the protein, you have to purify it, and then you have to combine it with an adjuvant. These typically can be stored and are stable in refrigerated temperatures. Their mechanism of action, you first have to identify the protein of interest from a pathogen. Once you've got that, you produce the protein in bacteria, yeast, or mammalian cells and combine it with an adjuvant and give it to the patients. Typically, they don't generate very strong T cell responses, but induce good antibodies that the adjuvant drives. Viral, while not relatively new, there's now a couple of viral vector COVID-19 vaccines that are approaching use or in use, including Sputnik 5, J&J, and Oxford AstraZeneca vaccines. What these vaccines do, they take a common cold virus called adenovirus, and they put the spike protein into the genome of the adenovirus. So when the virus infects a cell, it makes spike protein. The immune system recognizes that as foreign and makes an immune response. The research for viral vectors dates back about 40 years. There are a number of vaccines that are currently approved, including dengue virus and Ebola virus vaccines. The immunogenicity is variable. A drawback is that since you're delivering a virus, you also make responses against the viral vector in addition to the spike protein. And if you've already got immunity against that viral vector, the responses are often poor. It doesn't require handling infectious pathogens. There is a slight risk of chromosomal integration into the DNA, and there is risk with immunocompromised patients. They're typically given by intramuscular, although other routes have been studied. Large-scale culture production is required and has a medium pace of development. And typically they're stored at refrigerated temperatures. Their mechanism of action is similar to attenuated vaccines. They infect the cell. The viral proteins are produced from the RNA of the cold virus that induces antibodies and cell-mediated immunity. DNA vaccines have been studied for many years, but currently there are no approved human vaccines using DNA. They elicit good T cell and B cell responses, although the levels are variable and typically they're low potency, which means high doses, high amounts of DNA are required. There is a risk for chromosomal integration that could lead to cancer-causing mutations, although this has been found to be very rare. It doesn't require any biosafety issues and overall has a very safe profile. People use things like gene guns, which shoot particles covered in DNA into cells, or electroporation to improve potency. And manufacturing is very fast and very easy. And these vaccines are often stable at room temperature. The mechanism of action are that the DNA is taken up by a cell. The issue here is that the DNA has to cross into the nucleus. And that typically only occurs when the cell is dividing. And that's why things like electric shock are used to stimulate the cells for the plasmid to enter the nucleus. Once in the nucleus, RNA is made off of the plasmid, off of the DNA, that is then translated into proteins that stimulate T and B cell responses. mRNA vaccines are the sort of forefront for COVID-19 vaccines. The first two approved vaccines for COVID were both mRNA based. The heritage of RNA vaccines, they've been studied for 15 years. Um, People are nervous when they hear about the RNA vaccines because they say, oh, well, they were developed in under a year. Are they safe? They were developed in a year for COVID-19. They've been studied for 15 years for making vaccines. So this is not new technology. They have very favorable immune profiles. They induce very strong T cell and antibody responses. They have very high potency. The RNA is not infectious. It doesn't integrate into the cell. The RNA is encapsulated in a fatty droplet known as a lipid nanoparticle that allows easier delivery to cells in a person. Typically, the vaccine is given in 2 doses by the intramuscular route. One of the issues out is that it requires frozen storage. Both the Moderna and Pfizer vaccines require standard freezer temperatures and will last weeks to months at that temperature. The advantages of RNA are that they allow very fast development. All of the earlier vaccines I discussed required isolating the virus, growing the virus up in cells, or identifying proteins in the virus, cloning them, and growing them up in cells. With RNA, you only need the sequence. So last January 12th, 2019, 2020, when the Chinese released the sequence of the COVID-19 virus, we and others took that sequence and started to make RNA vaccines. We didn't need the virus. We didn't have to grow the virus. We just needed the sequence of spike, which makes it very fast to respond in urgent situations. The way the mRNA works is that the first thing you have to do is you have to choose what protein from the virus you want to encode. That's the spike protein. That can be easily plugged into an mRNA. There are other structures in the RNA that promote protein production. The mRNA is then encapsulated into a lipid nanoparticle. Those are endocytosed. What that means is that the cell essentially eats them. The RNA releases, it's translated. So essentially machines in the cell read the code of the RNA and make proteins. The proteins are processed to activate B cells and T cells, resulting in very potent responses. The RNA only lasts for a short amount of time, typically hours to a day or two in a cell. So you don't get long-lived protein production. And the RNA is degraded by natural cellular mechanisms. The RNA is identical to the RNA already in the cell. In addition, the RNA doesn't go into the nucleus. It can't affect DNA. It can't change DNA. It can't mutate the DNA. There are a number of steps when dealing with an RNA vaccine. So there are many elements. The first is choosing the antigen and preparing a DNA template. The way RNA is made is you start with DNA, just like the cell does. The DNA contains the coding sequence, so the sequence that allows the cell to make the protein. You put that into a construct that has other regions that were selected to give high levels of protein production. The RNA has other elements. One is a cap, the other is a poly A tail. These are used to allow the ribosomes, the machines, to read and make protein. The RNA is then encapsulated into the fat droplet, the lipid nanoparticle, which are also optimized for uptake by cells and for release of the RNA into the cell. There are numerous different kinds of RNA vaccines. There are unmodified, so these use the same 4 bases or letters in our own RNA. There's nucleoside modified, which is what Katie Carrico and I developed and are used in the Moderna and Pfizer-BioNTech vaccines. What we did is we added naturally occurring modified nucleosides. So these are modified letters that are found in regular RNA. And we found that when we incorporated those into the mRNA we produced, it got rid of the inflammatory potential. So it made the RNA much safer, and it allowed the RNA to make much more protein. The last type are self-amplifying RNAs. These are based on viruses that have figured out how to replicate, how to copy their own RNA. They tend to be very potent because if you put one RNA in the cell, the RNA has proteins that copy itself, making many RNAs, leading to high levels of protein production. So there are a number of late-stage COVID-19 vaccine platforms. 2 of them are already approved. Let me just discuss some of the drawbacks or potential issues or concerns of the different viral platforms. The principal one that people ask about is it infectious? Can you get the disease from the vaccine? That's a concern we hear all the time. None of these vaccines, viral vector, RNA, DNA, or protein-based, can give you COVID-19. It's impossible. The viral vectors can be alive and can, in rare circumstances, depending on the vaccine, can replicate and spread in immunocompromised hosts. RNA, DNA, and protein-based vaccines are not infectious. The viral vector vaccines and the DNA vaccines have the potential to integrate, to alter our DNA. RNA and protein-based cannot do that. There's the risk of an allergic reaction. So viral vectors, anything produced in a mammalian cell culture, can potentially do that. So that includes viral vector and protein-based. DNA and RNA do not use any mammalian cells or any eukaryotic cells and are very safe. The storage is variable. Most are able to be stored in refrigerated temperatures. The RNA vaccines currently require freezer temperatures, although the Moderna vaccine is stable for about a month in a fridge. The Pfizer vaccine is stable about a week in the fridge. So our conclusions. There have been multiple new standard and vaccine platforms that have been rapidly developed for SARS-CoV-2, COVID-19 vaccines. Newer platforms are generally genetic. What that means is that the protein in the vaccine is encoded in an RNA or a DNA. Genetic vaccines offer faster development, easier production, and easier change. So what that means is everybody's heard about these variants that have come out. To make a new variant RNA vaccine, you simply have to change the coding sequence with a variant coding sequence, and you've made a new vaccine. You don't have to isolate, grow, culture the virus. It's very simple and very fast. And let me stop there.
Thank you very much, Dr. Weissman. That was incredibly informative, and I learned a lot from there. We only have time for one question. So I'm going to ask one from Dr. Wamai. Um, he says, obviously, with COVID-19, the scientific challenge to develop a vaccine today has dramatically reduced in terms of the time frame. As we know, one of the reasons expressed by those who are hesitant to take the vaccine is the quick turnaround of under a year. And you've mentioned that this vaccine has been studied for a long time, for about 15 years, mRNA vaccines in general. So is the 1 year development timeframe just a problem of communication, or is it something else? And at any rate, it seems critical that the scientific community involves the stakeholders, which includes the public, in the scientific project of vaccine development to ensure like an early buy-in. And what's your opinion on this?
So I completely agree. I actually kind of think it's kind of funny because had we taken 2 years to develop it, they would have yelled at us, why is it taking so long to make a vaccine? So we're not going to win this conversation. What people have to understand is that nothing was cut short. The reason why most vaccines take 5+ years are there are 3 phases of clinical trials. They're spread out over years. You do a trial, you wait, you do the second, you wait, you do the third, you wait, you figure out how to make it, and then you sell it. With COVID, everything was done at the same time. All of the trials were done at the same time. They started making the vaccine to give to people before they were even approved, which is really shows a sign of pharmaceutical companies willing to take the risk. So while it was sped up, the size of the trials was the same as every other vaccine trial. All of the trials, all of the toxicity, all of the testing was identical. It was just done at the same time. And that's what needs to be messaged to people.
Okay, that's a really good answer. For other participants, other attendees, we do have some workshop sessions happening right now. You can find the links over at our website, and they'll be going from now until the end. Dr. Weissman, I actually have a couple more questions for you on like, because I have an interest in biochemistry, and I was just wondering the role of A.R.B. development in terms of the trying to target essentially COVID response in general, because there was some research published in the beginning that the coronavirus used the ACE2 receptors, and they thought of using ACE2 as a potential therapeutic. And then they found out that that wasn't necessarily effective. So I haven't had much time to look into that. I was wondering if you knew a little bit more about it.
Yes, we actually use ACE2 inhibitors as a hypertension drug.
Mhm.
So the early studies looked at the people taking ACE2 inhibitors do better. And they found that they didn't. So the issue is that the coronavirus infects respiratory epithelial cells in the airways. So the surface of the cell that's in the airway. So it's likely that an ACE2 inhibitor isn't going to reach that space. Even if it does, it's an affinity problem. And it's likely the spike binds better than the inhibitor, or the inhibitors don't block spike binding.
Okay. Right. That makes sense. Thank you. I think that's about it for questions. I think Fatima has some closing remarks, and then that's about it. Thank you very much.
Thank you so much, Dr. Weissman. Really enjoyed that talk. And again, thank you for being a speaker today. So this, this concludes the main session of the conference. And we will be moving on to our 2 concurrent workshops. Our first workshop will be Innovating in Global Health, where attendees will learn about the ACOA project and how they work with local communities to ensure safe and reliable medical services for all. And our second workshop will be the Health Literacy Workshop, Unraveling Health for the Public, where you will learn about health literacy and the principles of designing materials to communicate health information. We would like to thank everyone for attending day 1 of this conference, and I hope to see everyone in the workshops and tomorrow for day 2, where we will discuss the racial disparities that were exacerbated throughout the pandemic. Please go to the Zoom codes that you see on the slides here, or refer to the websites for the links. I look forward to seeing everyone in the workshops. Thank you.